Hello everyone, it's Ryan Maloney here at the National Jazz Museum in Harlem. Excited to have you with us for this virtual program today. On behalf of our board of directors, along with our executive director Tracy Hyder-Suffern and our artistic team of Jonathan Batiste and Christian McBride, we're really excited and welcome you here to another episode of Music on the Brain. Music on the Brain is presented in partnership with the Zuckerman Institute, our friends over at Columbia University, where they're doing all this amazing research on the brain and how it works. And this program is unique in that we pair a musician, in this case, the amazing pianist Helen Sung, with a neuroscientist, and they get a chance to explore the relationship between music and the brain. So to get things started, please welcome Helen Sung.
As you probably noticed, I played music which varied widely in tempo. And that is the theme of our program today, life, experiencing life, engaging with life at different speeds, different rates. And this is a large part of the research of our guest scientist, and I'd like to welcome her to the stage, as it were. Please give a big hand, you all out there, for our fabulous guest scientist, Sumaira Zamurrad.
Hello.
So one thing I love to do is, hear about your story, because I really think once you get past a certain threshold, science, I think most people, when they think science, they think numbers, they think very, precise results and kind of cold calculations. But actually, I think science, it is an art too. And I think there's a lot of cross relationships, points of commonality. So can you tell us a little bit about how you came to be doing what you're doing today?
Yeah, sure. I am currently now studying how our central nervous system controls walking, and I'll walk you through how I walked all the way here.
Nice.
So I'm originally from Pakistan. I was born and raised there, and then I came here for after high school for undergraduate studies, all the way from Pakistan to Stony Brook, New York.
Wow.
And so I did my bachelor's in biochemistry over there, and that was the first time that I was, got involved or even heard about research, scientific research. Growing up, my father's a physician, so I was always inspired by his work and was, drawn towards science. I was very curious, and so assumed that becoming a medical doctor would be my, my ultimate job. But I came here and, I'm really thankful to all of the opportunities and, the exposures I got to other fields in science as well, and started doing research with our biochemistry professor, Dr. Sanford Simon. And that's it. That just totally, opened a new door for me and made me realize that I was just really drawn towards scientific discovery and understanding how science works, and that's it. That sealed the deal. And thanks to those experiences and all my research mentors, I then started a PhD program at Einstein here in the Bronx, and that's where I started working ultimately with flies, Drosophila, studying intellectual disability using a Drosophila model in Dr. Julie Secombe's lab. And after that, I was at a fly meeting in Cold Spring Harbor, where I heard a talk from Richard Mann's lab. And that too was about walking behavior in flies, and that drew me here. And I talked to him, and that's how I ended up here at this amazing institute, surrounded by amazing colleagues and world-class neuroscientists.
Nice.
So.
Wow. Well, I know I've heard about the Drosophila in many different settings. And so can you share with us what you're doing with them? And
Yeah.
You have some cool slides to show us.
Let's use some slides. Okay. So as I said, we, in the Mann lab, we do a couple of different things, and one of them is studying walking behavior. And so, this is something that we all take for granted oftentimes. We just walk everywhere without really realizing that there is a whole complex system of organs and neurons in humans, in our bodies, that's regulating every aspect of every behavior, in fact, but for our behavior of interest, walking. And so in humans, we have the brain connected to the spinal cord, which then connects via ligaments, tendons, muscles, neurons to all our four limbs, and that's how the brain is controlling all our motor movements. And this is just a sort of a zoom in, showing you this complex circuitry between the brain, spinal nerve cord, and our limbs via sensory neurons and motor neurons, and how this forms this complex, constant loop of information, which is taking whatever we see, we feel, we sense down via neurons to our ventral nerve cord, spinal cord into our limbs, and then what we sense from our environment, from our limbs, and taking it again back to the brain. And so we have this constant back and forth of information that is allowing us to regulate how we complex our ever-changing environment.
Yeah.
And so in the Mann lab, what we do is, trying to this circuitry. We know that it exists. We know in broad terms that these neurons exist, but there's a lot more of those intermediate neurons called interneurons, et cetera. And even ultimately, how these neurons connect to motor neurons, what the circuitry is controlling each aspect of each precise behavior still remains unknown. And that's what we're trying to unravel using various tools and techniques. And the model organism that we use to ask and answer these questions is Drosophila, commonly known as the vinegar fly. And so, and you might ask, why would you use a fly to study this something, and whether it's actually applicable to humans? And so what's really interesting is that there is a lot of genetic similarity between flies and humans. So it's about 60%. So about 60% of the genes that are found in humans associated with a specific output or function also exist in flies. And so it's a really great, very useful, cheap, economical model organism, which also reflects the genetics of humans, and so we can ask some very basic fundamental questions about the functions that those genes carry out. And so also, as you can see, if you just go back one slide, the most important question is that the central nervous system in flies, you have the brain and the ventral nerve cord, is analogous and similar to how it's laid out in humans, where again you have a brain connected to a spinal nerve cord. And so again, we see that the fundamentals are similar. The genetics there's a lot of similarity in the genetics, and that will hopefully convince you that this is a great model organism to ask those questions in. And again, in the flies too, the ventral nerve cord is connecting 6 jointed limbs. And that's what happens in humans as well. Your spinal cord is connected to your limbs, and that's what's controlling the limbs. But in flies, in addition to the limbs, you also have a pair of wings. So all in all, far more complex if you think about it.
Yeah, would you say flies are smarter than us?
You would think, because they're doing way more, right?
Amazing.
Yeah. So this is just a zoom in. Just trying to show you that we're trying to focus in on a specific set of neurons called the descending neurons, and this is just a schematic showing you a blow up of the CNS, where you have the brain and the ventral nerve cord. And these descending neurons are a very important primary group of neurons that originate in the brain, where they have a cell body in the brain. And then in the brain, they're receiving all this information from various stimuli, from their eyes, they're sensing information, and they're sending it down to the ventral nerve cord. So they're sort of the primary conduits of information, and they act as a bridge of carrying the information from the brain to the ventral nerve cord.
So do flies smell too? I mean, what kind of,
yes.
They feel wind, they feel...
Exactly. Mm-hmm.
So the descending neurons take that information and send it down.
Various descending neurons are able to, this is just showing a descending neuron with a cell body in this particular region of the brain. There's connecting to different regions of the brain, and depending on where they're originating, they're going to carry information that region is specialized for obtaining and then carrying it down.
Nice.
And again, relaying it to the important, parts of the VNC, which can then be carried to its precise appendages.
Okay. Wow.
And so the question we know that these descending neurons are important in carrying out specific behaviors, but again, they are at the very top of the circuit. And what are they connecting to in the VNC? Obviously, they have multiple partners in the ventral nerve cord. What are those partners? Which one of those partners are connecting to which one of the legs or appendages or wings? And how those specific partners are then controlling walking behavior is still unknown. And so that's what we're trying to use different techniques and tools to try and understand.
Cool. Did you?
So like I just said, this is the goal of our work is ultimately to try and map out this, unknown, unraveled circuitry, and then try to associate those different circuit components to precise motor behaviors.
Well, thank you, thank you. And to bring the musical connection in, music is something that is, that definitely is something that the descending neurons must engage with in some way. Music is used to affect our behavior in some ways too. I think sometimes it's on purpose. I think that I was reading how stores pipe a certain type of music that can affect whether you shop faster or slower. And also, they said the most dangerous piece of music to drive to is the Ride of the Valkyries by Wagner, which I don't know if you guys know it. Because apparently it makes people go fast, and and I think that's really interesting because that just shows you how time can become subjective too. Like that's something that descending neurons make us press the gas pedal a little bit faster. But so I'd like to do that in a less deliberate way. But just I'm curious. I'm going to play some musical selections, and if you guys wouldn't mind sharing, like maybe we can think about just adjectives that come to mind, or maybe how it would want you to inspire you to move. Think about movements that you might associate with each selection. So here's the first one. This is a piece by the great Marian McPartland. It's the theme song to her long-running NPR show called Piano Jazz. And the piece is called Kaleidoscope. So any thoughts, any response?
Hectic.
Hectic, okay. What about any movements that you might have associated or come into your mind, or that you might want to move to?
I feel like I was twirling around, being unmotivated.
Twirling around, nice.
I felt like walking upstairs very quickly, like trying to get somewhere. Perhaps having to stop and catch my breath a few times.
Yeah, no, that's great because the line is this ascending line, I think. So that's a great association. Thank you. This next piece I'm going to play is by one of the other giants of jazz piano, the great Mary Lou Williams, called "What Is Your Story?" What's Your Story? Morning Glory. Any thoughts? Any thoughts about that one?
That one made me think of, I'm familiar with, that song is familiar to me, and so whenever I hear it, I think of like someone kind of like walking slowly and like grazing against things gently, but there's like a confidence in it somehow also, that
Nice.
aspect of confidence. So
Okay.
just like carefree and just gracefully walking about.
Nice, thank you, thank you. Okay, I'll do one more. Let's see, what should I do? All right. Any thoughts? Let's ask our guest.
That made me want to dance a little bit, and I'm a very bad dancer. You know that.
No. Okay. What about it made you want to dance?
I think it was fun and lighthearted.
Hearted? Yeah. It's a great piece. This by the Gershwin brothers, George and Ira Gershwin, called "Nice Work If You Can Get It." So, yeah. So as you listen to those songs, as you listen to music, maybe this week as you're walking around, and just think about what your descending neurons might be doing. It's kind of interesting. I love it. So now you're actually, tell us a little bit more about these neural networks, right? Because when they work together, you call them networks.
Yeah. Okay. So, so like I alluded to before, we know that there's these descending neurons that are at the very, almost at the top of the circuit. They're command-like neurons that are receiving the primary set of information in the brain, and then they talk to other downstream neurons that come after them, eventually going into the limbs. But we don't really know those neurons, and there's many hypotheses about how these neurons at the top may be talking to the ones at the bottom, called the downstream neurons. And it could be very simple as one neuron talking to the second one, then talking to the third one. So you have primary, secondary, tertiary. Or it could be a bit more complex where, like in the middle, you see one neuron talking to three, then those three are talking to another set of three neurons. Or you could have one neuron talking to two different neurons at the same time, which are then talking to three different neurons at the same time. And so, you can have these increasingly, progressively more complex neuronal networks, so to speak. And depending on the kind of information that they're carrying, or the kind of information that they need to relay to possibly different sets of limbs or ultimate partners to control certain behaviors, you could imagine the complexity just keeps on increasing. And so this is what we are trying to ask is, what do the descending neurons, especially the ones for which we have some information about that they are potentially regulating walking behavior, what are their downstream partners? Who are they talking to? And what on what sort of level of complexity, or how many neurons are they talking to, or exactly what they're doing? Or is there some sort of like intermediate important neuron, which is then relaying information to another important set of neurons called the motor neurons, which, as the name implies, control motor movements. Is it something like that? So we have no idea. We have only like hypotheses, and thanks to lots of tools and techniques, including behavior assays, imaging, we are starting to unravel and answer these questions. At least we're starting.
I'm just stunned because things like you said, I think you said at the beginning of this program, that these actions, these movements, we do almost unconsciously. And to really, when you're drilling down, just looking at something as basic as walking, it's unbelievably complex. So it's just, that to me is why I love doing these programs, because it's a celebration of our brains and just the amazing things that it allows us to do, enables us to do.
Exactly. Yeah, and so if you go on this slide, what I'm just trying to show you is one of the really cool techniques that we used, many groups have used and we also use in the lab, called optogenetics. And as the name implies, it allows us to use specific colored light to activate specific genes. And we have the tools to manipulate every single, or one single gene controlling a single neuron, that it will then respond to the light, and only it will respond to the light. And so what that allows us to do is say one neuron in a circuit is, and manipulate just one neuron in a circuit and ask if you do that, do you see a change in behavior? Is there a specific neuron associated with a specific behavior?
So how do you go about deciding which neuron? Is it like it's an educated, not guess, but a choice because of other things leading up to that particular neuron, or do you just kind of going down the line?
It's sort of a forward screening approach where you start, like I said, with the descending neurons, which are pretty much at the top of, obviously there's more neurons that are upstream of the descending as well, but there are a main set of command neurons, and you sort of like this paper has done, is where they just started by screening those. They just took each descending neuron, activated it like it's shown over here, with using red light, and then they just basically videotaped them and saw what happened to the flies when you activate that descending neuron. And if we go to the next slide, I think we can see an example of what happens when so in this fly you have one descending neuron that has been manipulated such that it will respond to red light.
Oh wow!
And so when the red light comes on, you'll see the red light. It'll be labeled at the bottom, and you'll see that the behavior changes slightly. Let's see if you can.
Oh wow!
If you can see, so it was walking with all 6 legs, but as soon as the red light comes on, it switches to it actually stops and switches to using its front 2 legs to start cleaning its eyes, grooming.
And so it's consistent.
Yeah, exactly. And so when you can turn off the light, it'll stop. You turn on the red light, it'll start. So it's really cool that some of these, this broad screening approach reveal that similarly that some descending neurons are really important in behaviors like grooming, some in fast walking, some in slower walking, some in freezing, slowing down. And so that was the first set of information that has revealed that some of these descending neurons are playing an important role in specific kinds of behaviors.
Is there redundancy though also? Like, is there more than one of these neurons that would
For sure.
do the same thing?
I'm sure there are many neurons that are controlling similar behaviors, and that might be just important for survival.
Yeah.
But then there might be nuances. You might have specific kind of grooming behaviors. You might be grooming with one leg, or you might be grooming with the other, or you might be doing a specific kind of grooming behavior that covers part of your eye and not the other.
Makes my head spin.
Yeah.
That's amazing.
And this is just another example of a similar, another descending neuron, which when it's activated using red light, you'll see that it starts walking backwards. So that's the left panel. You can see that that fly is constantly walking backwards when this specific descending neuron, called the Moonwalker descending neuron, is activated using red light. So
it's like they
pretty
almost
fascinating.
can't help themselves. Like, I wonder if it's thinking, what is going on?
Yeah, exactly. They're just, because
Wow.
I mean, typically you'd walk backwards if you have some sort of something approaching you
Right.
that you're trying to,
Avoid.
Well, and fly could fly away, but obviously it's capable of walking backwards.
Amazing.
Um,
Amazing.
So that's just some examples, but we have other examples where we can see that when certain descending neurons are activated, you can see an obvious increase in the velocity or the length of time that the fly will continuously walk for, or it'll just or it'll walk slower. And so that's really, I think it has really educated us. It's been very informative in helping us decipher which ones to study further. But again, this is just the very first set of command neurons. The real question is, what are the downstream neurons in the ventral nerve cord? What are the neurons in the circuit that ultimately reach the leg and then control it to do this, to walk slower or faster or stop or freeze, and all of that.
Fascinating. So I would love to hear a little bit more about this other tool, very powerful tool, very new tool, I think, right?
Yeah.
Is it, how many years old? Like,
It's very recent, like in the past few years.
wow,
Yeah.
wow.
Well, the technique is not new, but this data set is very new that
okay.
it's been done for the first time for the
Because
the
it
fruit
hasn't been
fly.
able to be done at this fine a level, or?
Or not for the fruit fly, it has not been done. Yeah.
Oh, okay, okay.
And for this particular
Got
organ.
it. Okay.
So another way, one way is to obviously, one question is to ask what descending neurons are responsible for what behaviors. But again, like I alluded to, we don't know those neuronal circuits that are downstream of these descending neurons that ultimately reach the legs. And one way of trying to understand is, there have been many other techniques that allow us to label those downstream neurons, but we can also recently, this data set has been published called, so what they did was they used electron microscopy to uncover, so basically to image the whole ventral nerve cord at very high resolution, so that we can see all of the neurons and trace them to really see the neuronal circuits in the ventral nerve cord. And so over here you can see that what the dashed box is showing the ventral nerve cord that they then used to basically use in electron microscopy to image, and they basically made about 5,000 longitudinal sections of this very tiny little ventral nerve cord.
Longitudinal meaning if the ventral nerve cord is like this, slicing like this.
Yeah, because it would
Oh my
be
goodness.
less work. Because if you went like this, it
Right,
would be like way more.
right. Yeah, true. Well, do nerves travel? They normally travel like
Yeah.
that anyway, right?
Yeah. But it doesn't matter whether you do it this way or that
Oh,
way,
really?
because once you put it all together, you can, your
true.
image is the same.
True.
Your
Okay.
3D image is the same.
Got it,
Because
got it.
that's the ultimate goal, is that you have 5,000 sections. Can we just go back to the previous slide? Thanks. So you have 5,000 sections of this ventral nerve cord, and then you put them all together and you have this nice 3D image of
Amazing.
ultrastructure,
Wow.
substructure. And this is just showing one section.
That's the 3D one.
Yeah, this is just a 3D image
Yes.
of the ventral nerve cord, and that's one section that was imaged, and
Oh my gosh.
that's a zoom in of what you would see of that little green boxed area.
Oh, wow.
And there you can see the potentially the neurons, and then we know what neurons look like, or what neurons are pre or post synaptic, meaning which one is upstream, and there's specifics like things like vesicles that are found in presynaptic neurons that you can help us map them. Like you can see which one is the next partner of this upstream neuron, and basically trace the whole circuit.
What is a vesicle?
Vesicle is a very small structure that contains neurotransmitters, or that's what the neuron is
part of the neuron.
transmitting. It's
Oh,
like
okay.
the information. Yeah,
Oh, I
that's
see.
the information that it's transmitting
Amazing.
to the next neuron.
Okay.
Um,
So you're
So-
basically mapping out little by little, or you're following a network down to where you think
the
it's-
ultimate target, ultimate motor. So
Okay.
hopefully, like for walking, it would be motor neurons.
Yes.
We would think, and so, but we need to know what lies between descending neurons and motor neurons
Yes.
potentially,
Yes.
and those are interneurons. And then, are there like is there a very important interneuron in the middle that is critical to receiving information and then transmitting it to all these motor neurons, or a group of motor neurons for a specific walking behavior.
I don't know, this makes me think of when I was in school, the atom was the smallest particle in the universe, but then now I don't think it is anymore, and I don't know, do they study, I guess, neurons themselves are made up of things that, right?
Yeah, neurons are very complex structures, they
Amazing.
have, and if you go to the next slide, we can, there's an example of, so this is showing what we've learned from that EM study. We're able to, so the green is an example of a descending neuron that we're interested in. And the one in, what is this, pink or orange, the contrasting color is a motor neuron. And we know that these two are directly connected, especially at those little yellow points. And so,
Wow.
this is just one of many of hundreds of targets of this descending neuron. And, and in this example, it's connected directly to a motor neuron. But that's not very, that doesn't happen very often. Oftentimes you have lots of neurons in the middle.
That's a great find.
That is a very interesting find.
Wow.
So yeah, this tool is really helping us, and thanks to our collaborators who published this data set about a year or two ago, where we've really been able to use this resource to advance our understanding of these neuronal circuits.
How long do you think it takes to map out a complete circuit?
Yeah, so this is becoming faster because I think in the past 2 years at least, it was being done manually, where you had to go to each neuron, find the next one, and manually tell it this is the next. But now, thanks to artificial intelligence and machine learning, they've paired up with a company that has paired this. You can teach a computer software to find the next target. But that is not. Yeah, exactly.
So using those 5,000 slices.
Yep.
Oh boy.
And
Wow.
so it can do, you basically pick one and then it'll map it all the way, hopefully, but then sometimes it makes a lot of mistakes. And so humans do have to go in and proofread it.
So I guess you still go in sections, but it goes a lot faster.
Yeah,
Okay.
exactly. Yeah.
Brilliant. Wow. You're making my neurons spin inside, Sumaira. Let's close this program like we usually do with music, and I'm going to play a piece by the great Chick Corea, a piece he wrote for his dad called Armando's Rumba. And I'm going to play it in many different styles, as many as I can think of while I'm doing it, and maybe just let the music. Mike, should have brought your guitar. I'll play it in different styles, and just enjoy letting the music travel through your neurons, descending or otherwise. much again for joining us. I also want to thank my wonderful partners in this series and collaboration, everyone from the Zuckerman Institute, Dr. Paula Croxon, Alyssa Mayers, Marisol Estrella Vivas, and Ryan Maloney from the National Jazz Museum in Harlem, and of course, our wonderful guest scientist today, Dr. Sumaira Zamurrad. It is always such a pleasure to examine and explore the connections between music and science, and we hope to see you again at our next episode of Music on the Brain.